Assalamu alaikum. Welcome to Wetlek. Today the topic of discussion is glands and their secretions. So first I am discussing the salivary glands. The salivary glands are actually exocrine glands that produce saliva through a system of ducts. These salivary secretions originate in the gland acini and are modified in the collecting ducts. Salivary secretions are regulated by parasympathetic nervous system. Now come to the composition of the salivary glands saliva. So we see that ruminant saliva is alkaline in nature and is a bicarbonate phosphate buffer. The function of this well buffered saliva is for neutralizing the acids which are found by the fermentation. As we have seen that the pH of rumen is from ranges from 5.8 to 6.8, and rumen also undergoes fermentation and produces acids. So in order to neutralize these acids found by fermentation, this well buffered or alkaline saliva is necessary for neutralizing these acids. Saliva also contains antibacterial and antimicrobial enzymes which are known as lysozymes. And the function of these lysozymes is to destroy the bacteria and protect the oral passageways or oral cavity from bacterial invaders. The saliva of omnivores contains salivary amylase, whilst this salivary amylase enzyme is absent in carnivores. The function of this salivary amylase is actually in the digestion of starch. As carnivores eat protein rich diet, so they don't need salivary amylase for the digestion of starch, as they don't consume starch. So due to protein rich diet, carnivores don't need salivary amylase. Another enzyme known as lingual lipase is present only in young calves when they are on milk diet, and this disappears when they become mature. This lingual lipase is secreted from the serous glands which are present in the tongue, and it functions in the breakdown of triglycerides into fatty acids and glycerides. This breakdown of triglycerides into fatty acids and glycerides occurs in the stomach, and actually this conversion is catalyzed by this lingual lipase. So we can say that lingual lipase contributes to the hydrolysis of dietary fats in the stomach of mammals. Salivary gland is typical acinar gland. An epithelium of acini secretes saliva into the lumen of acini. Then the glandular cells, which are also present in the walls of acini, secrete water, electrolytes, mucus, and enzymes. As the saliva progresses through the collecting ducts, its composition becomes modified because the epithelium of the collecting ducts reabsorbs its electrolytes, and it absorbs sodium and chloride. So the final product is the hypotonic saliva, with a concentration of sodium less than that of extracellular fluid. Let's repeat it again. Epithelium of acini secretes saliva into the lumen of the acini. Then glandular cells secrete water, electrolytes, mucus, and enzymes into the lumen. Then the epithelium of the collecting ducts absorbs the electrolytes, and the final product is hypotonic saliva. And this hypotonic saliva has a concentration of sodium less than that of the concentration of sodium in extracellular fluid. There are actually three types of salivary glands: parotid glands, sublingual glands, and submandibular glands. Parotid glands are present under the ears or behind the vertical ramus of the mandible. Sublingual glands are present below the tongue, and submandibular glands are present in intramandibular space. Cattle and buffaloes produce 130 to 200 liters of saliva per day. Goats and sheep produce 2 to 3 liters of saliva per day, and horses produce 10 to 12 liters of saliva per day. Now let's talk about pancreas. Pancreas is actually a dual gland. It has both endocrine and exocrine activity. The exocrine activity is the major activity of pancreas. So pancreas has 99% exocrine activity and only 1% endocrine activity. So pancreas is also an acinar gland, and it comprises acini, lobules, and centroacinar cells. Its exocrine portion contains acini, lobules, and centroacinar cells, and acini and lobules secrete pancreatic juice. Its acinar cells also secrete enzymes, while centroacinar cells, which are spindle-shaped cells and are present at the extension of the duct into the pancreatic acinus, secrete sodium bicarbonate solution. And this aqueous sodium bicarbonate solution is secreted under the stimulation of the secretin hormone. The receptors of pancreatic cells are stimulated by secretin, acetylcholine, and cholecystokinin. And when these receptors get stimulated, then the acini of pancreas secrete pancreatic juice. There are canaliculi also present between the acini, and these canaliculi between the acini unite to form small canals among lobules. And these canals of lobules then form larger ducts, and these larger ducts then unite to form a single pancreatic duct which opens into the duodenum. But before opening into the duodenum, main secretory duct of the pancreas fuses with the common bile duct which is coming from the liver, and these both then unite to form major pancreatohepatic duct. And this major pancreatohepatic duct opens into the duodenum through ampulla of Vater, which consists of sphincter muscles for the prevention of the duodenal contents to move into the pancreas. In this way, it controls pancreatic secretions. Let's repeat it again. Canaliculi, which are present between acini, they unite to form small canals among the lobules of acini. Then these smaller canals unite from and of the in the one lobule and then form larger duct. Then these larger ducts from different lobules unite to form pancreatic duct. Then this pancreatic duct fuses with common bile duct from liver, and then pancreatic duct, pancreatic duct, main pancreatic duct, and the common bile duct fuse to form a major pancreatohepatic duct. This pancreatohepatic duct then opens into the duodenum through ampulla of Vater, and this ampulla of Vater contains sphincter muscles for the prevention of duodenal contents to move into the pancreas. The enzymes of pancreatic juice require an optimum pH of 6.9 for their action in the small intestine, while the material that comes from the stomach into the small intestine is acidic in nature. So pancreatic juice is secreted at the duodenum, the very first portion of the small intestine, and this secreted pancreatic juice then optimizes the pH of the small intestine to 6.9, and in this way it allows the pancreatic enzymes to work properly. As compared to monogastrics, in ruminants, the pancreatic juice of ruminants contains a relatively low bicarbonate content. And due to this relatively low bicarbonate content, the pH of the acidic digesta that is entering the duodenum increases only slowly. Pancreatic lipase, as its name indicates, that it will act on the lipids. So pancreatic lipase causes hydrolysis of fats into carboxylic acids and glycerol, and the action of pancreatic lipase is facilitated by the prior emulsification of fats, which is caused by the action of bile salts in the liver. As the fats are first emulsified by the bile salts contained in the gallbladder of liver, and in this way the action of the pancreatic lipase is facilitated by the prior emulsification of fats. So the complete hydrolysis of fats in small intestine is accomplished by the combined activity of both bile salts and pancreatic lipase. In adult animals, the activity of pancreatic lipase increases when the animal consumes high grain diet. In most farm animals, bile and pancreatic secretions are carried into the duodenum through the common major hepatopancreatic duct. While in case of cattle and buffalo, the pancreatic juice and bile are discharged into the duodenum through separate ducts. Now let's move to the composition of pancreatic juice. Pancreatic juice contains 98.55% of water and 1.5% of solid content. This solid content is again divided into organic constituents and inorganic constituents. The organic constituents include proteases like trypsin, chymotrypsin, collagenase, carboxypeptidase, and elastase. Then other organic constituent is the amylolytic enzymes, which are pancreatic amylase and amylopsin. Then the other organic constituent is the lipolytic enzyme like pancreatic lipase and strypsin. The other organic constituent is deoxyribonuclease, phospholipase, and cholesterol ester hydrolase. The inorganic constituents of the pancreatic juice are sodium, potassium, calcium, magnesium, iron, chlorine, and traces of other elements. As I have told you that pancreatic juice is highly alkaline in nature, so it has a pH of 8.3, and this high pH is due to the abundant content of bicarbonates in the pancreatic juice, and in this way it maintains alkalinity in small gut. Pancreatic juice digests proteins, carbohydrates, lipids, and other food ingredients. So the proteases present in the pancreatic juice cause the digestion of proteins. Amylolytic enzymes that are present in this pancreatic juice cause the digestion of carbohydrates, and lipolytic enzymes cause the digestion of lipids. Now I am going to talk about the regulation of pancreatic juice. Pancreatic juice secretion is regulated by both sympathetic and parasympathetic nervous system. The secretion of gastrin from the duodenal mucosa has a major role in the regulation of pancreatic juice. Distension of the stomach nerves also stimulates the secretion of pancreatic juice, while vagotomy and atropine inhibit the secretion of pancreatic juice. Secretion of secretin from duodenal mucosa also causes secretion of pancreatic juice, and this juice, which is released by the we can say stimulus of this secretin, is rich in bicarbonate and deficient in enzymes. Duodenal mucosa also secretes cholecystokinin or CCK-PZ, and then CCK-PZ then causes the secretion of pancreatic juice, which is rich in enzymes. The major secretors of pancreatic juice in the duodenum are CCK-PZ or cholecystokinin and secretin. Now let's talk about the enzymes which are present in pancreas. Enzymes which are present in pancreatic juice. There are two types of enzymes present in pancreatic juice, which are exopeptidases and endopeptidases. There are also lipases and amylases and other enzymes, but in case of peptidases, there are two types of peptidases which are present in pancreatic juice. One is exopeptidases and others are endopeptidases. Exopeptidases include trypsinogen and chymotrypsinogen, while endopeptidases are pro-carboxypeptidase and pro-aminopeptidase. Then fat and lipid digesting enzymes which are present in pancreatic juice are lipases, and these include phospholipases and cholesterol esterases. The carbohydrate digesting enzymes of pancreatic juice are pancreatic amylases, and the DNA and RNA digesting enzymes of pancreatic juice are deoxyribonucleases for DNA and ribonucleases for the digestion of RNA. In case of exopeptidases, we have discussed that there are two exopeptidases which are trypsinogen and chymotrypsinogen. So these both trypsinogen and chymotrypsinogen are actually the inactivated forms of trypsin and chymotrypsin. So trypsin and chymotrypsin are activated forms of trypsinogen and chymotrypsinogen, and these both are activated. Both trypsinogen and chymotrypsinogen are activated by special enzymes which are called enterokinases and are present on the surface of duodenal cells. So when these enzymes trypsinogen and chymotrypsinogen come along with the pancreatic juice through pancreas through major hepatopancreatic duct into the duodenum, then they are exposed to the enterokinases, and then these enterokinases will convert trypsinogen into trypsin and chymotrypsinogen into chymotrypsin, and then these activated forms will act on the proenzymes and will activate them. Now let's move to the gastric juice. Gastric juice is actually a colorless acidic fluid which is secreted by the glands of the stomach, and it promotes the digestion of food. So this gastric juice is formed by the various glands of stomach, and this gastric juice is slightly hypotonic than the blood plasma because it contains 99% of water and only 1% of solids. These 1% of solids is again divided into organic solids and inorganic solids. Organic constituents of the gastric juice include pepsin, mucin, gastric lipase, intrinsic factor, lactic acid, carbonic anhydrase, rennin, urea, and urease. On the other hand, inorganic substances include HCl, hydrochloric acid, sodium, potassium, phosphates, and other trace minerals. So the organic substances which are present in the gastric juice play very important role in the function of gastric juice. For example, and inorganic substances also does the same. For example, hydrochloric acid softens and hydrolyzes the food, and it also performs acid antiseptic actions because it kills the microbes. And when it kills the microbes, this hydrochloric acid makes their proteins available to the body. Hydrochloric acid also assists in absorption of iron by the conversion of ferric into ferrous. So hydrochloric acid performs many functions. I am repeating those functions. First function is it that hydrochloric acid softens the food, then it hydrolyzes the food, then it performs antiseptic action and kills the microbes. When it kills the microbes, it makes their proteins available to the body, and last function is that it assists in the absorption of iron by conversion of iron from ferric form to the ferrous form. Then the other organic constituent in the gastric juice is pepsin, and pepsin is very important protein digesting enzyme, which is activated form of pepsinogen. Pepsinogen is activated only in the stomach where it is activated into pepsin on its encounter with hydrochloric acid which is present in the stomach or already activated pepsin. So pepsinogen is only activated in the stomach when it encounters hydrochloric acid or already activated pepsin. In this way, pepsinogen is activated to pepsin, and this pepsin causes protein digestion. The other organic constituent of gastric juice, we have discussed that it is gastric lipase, and gastric lipase, as the name its name indicates, lipase. It hydrolyzes fats, and main function of gastric lipase is to digest milk fats and cause egg hydrolysis. The next organic component of the gastric juice was mucin, and mucin is its word its name indicates that it is related to mucus. So mucin actually lubricates the stomach walls, and by lubrication it protects the wall by the action of hydrochloric acid and pepsin on it. And so it protects the wall by forming a layer of mucus on it, and in this way it acts as a buffer medium. In the pyloric region of the stomach, there are patches of lymphoid tissue, and these patches of lymphoid tissue actually provide the immunity. And in addition to providing immunity, they also function in the absorption of short chain fatty acids and VFAs, that is volatile fatty acids, from the stomach. In this, similarly, intrinsic factor from parietal cells is necessary for vitamin B12 absorption. So this intrinsic factor is actually a glycoprotein, and it is produced by gastric parietal cells, and it enables the body to absorb vitamin B12. Now we see that how gastric juice is regulated or how its production is regulated. So there are three phases of the regulation of gastric juice. These are neural or cephalic phase, gastric phase, and intestinal phase. In the neural or cephalic phase, stimulants of gastric juice like smell of food, its sight and taste of food cause the stimulation of nervous system, and in this way nervous system causes secretion of gastric juice, and in this way it brings about the secretion of gastric juice. Similarly, sham feeding also causes the secretion of gastric juice. So what is sham feeding? Sham feeding is any procedure that mimics the normal food consumption but where food and drink are not actually digested or absorbed. Sham feeding is actually a experimental process which is used to study hunger, eating, or digestion in animals. If we does this experimental study, that is sham feeding, then this sham feeding also causes the secretion of gastric juice. Then comes the gastric phase. When the food touches the walls of the stomach, then it stimulates them to secrete gastric juice. So, in this way, in gastric phase, secretion occurs due to the touching of food with stomach walls, and this gastric phase is also known as chemical phase. After gastric phase, there comes the intestinal phase. The intestinal phase of gastric secretion involves the entrance of acidic chyme into the duodenum of the intestine, which inhibits the digestion process. And gastric secretions are this inhibition of gastric secretion is caused by the release of secretin into the bloodstream. I repeat it again. Intestinal phase is a stage in which the duodenum responds to the arriving chyme and moderates the gastric activity through hormones and nervous reflexes. The duodenum initially enhances the gastric secretion but soon inhibits it by the release of secretin into the bloodstream. 5 to 10% of gastric acid secretion occurs through this intestinal phase. So, when partially digested food fills the duodenum, intestinal mucosal cells release a hormone called intestinal gastrin. This further excites the gastric juice secretion, and this stimulatory activity is brief because when the intestine distends with chyme, it inhibits the secretion of gastric juice. So, the cephalic phase, which is the act of eating, the gastric phase, which is the arrival of food in the stomach, and intestinal phase, which is the arrival of food in the intestine, all these three phases cause the secretion of gastric juice. Now, let's talk about the substances that are stimulants and depressants of this gastric juice. So the stimulants are those substances that cause the stimulation of the production of gastric juice, means they increase the production of gastric juice. And these stimulants are histamines, caffeine, nicotine, insulin, alcohols, proteins, chilies, corticosteroids, anger, anxiety, and fear. While on the other hand, depressants are those substances that decrease the secretion of gastric juice, and these depressants are antihistamines, antigastrin, secretin, fats, alkalis and acids, parasympathetic agents, anxiety, pain, and fear.